Howard Hughes Medical Institute early career scientist. Works obviously in embryos. I'm an embryologist, and I actually think that we could just roll along because of your work will speak for itself. So
Absolutely.
This is great. Thanks a lot.
The previous speaker was both an epidemiologist and an internist, and I'm not anything that's nearly quite so impressive. But it's funny just listening to these talks. This is not the kind of meeting I go to very often, so it's been very fun. We've had sex, and we've had drugs, and death from all kinds of exciting things, and so I'm not exactly sure what I'm going to add, perhaps a bit of comic relief, but oh, we have reverb? Am I too loud? Oh, okay. All right, yeah. Well, so what I'm going to do is just tell you about what we do. We're on the absolutely opposite end of the scientific spectrum. We are just curious scientists asking questions about things that happen in biology that fascinate us, and really the story is kind of I hope a story about the promise of basic research, and also, I don't know, I just think it's really fun. And so I'm going to talk about sort of the work that my lab does. We use frog embryos generally, and we've been studying early embryological development. And then we sort of took a detour into the research into basic biology of the airway and airway pathologies. And I hope you'll enjoy it. I think it's been kind of a fun journey for me. So really what we're most interested in is the process called morphogenesis. This is how embryos acquire their shape. If you think about a chicken and an egg, they don't look anything like each other. Egg's pretty boring, roughly symmetrical, not quite spherical, but utterly boring on the outside. You dig inside, you'll see some interesting stuff. It's pretty boring. Chickens are actually a fairly interesting shape. Frogs are the same way. They start out as a little sphere like this picture here, and at some point they're going to make a tadpole like that. And the question that fascinated me for the last 20 years or so have been, how do embryos generate these shapes? How is shape generated in embryos? And the particular shapes that I've been interested in are the shapes in the frogs. So we do the vast majority of our experiments in the frog of a genus Xenopus. Now, not necessarily that many are going to have heard of Xenopus, but it's a widely used vertebrate model system. I think a lot of people probably know about zebrafish for basic biology research. Xenopus is the complement to zebrafish. You do many of the same things, and some things it does quite a bit better, some things it does not quite as well. It's a little less popular than zebrafish, but it's a vertebrate animal. We like it because it develops externally, which means we can watch it happen under the microscope, which for people who are image-oriented like myself, that's really important. The embryos are enormous, 10 times bigger than mammalian embryos, and so that makes them easier to manipulate and also easier to see. The cells are big. We have a sequenced genome. Amphibian embryos have been studied for over 150 years, beautiful papers from the 1850s on the formation of the nervous system in amphibian embryos, and we have lots of tools and tricks we can do. And so a quick disclaimer about this talk. There's going to be a lot of these pictures. They're pretty. 99% of the pictures, you don't even need to see anything in them. They're simply there to look pretty for you. They are actual data. If you're a developmental biologist or a molecular biologist, you might want to look more closely. But if you're not, don't worry about it. If you see a really long word, you can ignore it. It's there for the people who know what it is. If you see a gene name, you can probably ignore that, with a couple of exceptions, and I'll tell you about those. And please interrupt me at any point. I'm a biologist. I tend to speak in jargon. Please, if I say something and you don't know what it is, just yell at me, and I'll explain it. So, as I said, not that many of you know about Xenopus, but you probably should. It's been a hugely powerful vertebrate model system. So why do people work on Xenopus? As I said, amphibians have been studied for about 150 years. The problem was always you had to wander off to some pond, pull some embryos, some tadpoles out of the pond, hope they're at the stage you wanted, do your experiments. Well, then it was found that Xenopus was really nice because, pardon me for being crass, but you could take pee from pregnant ladies and then put it into the frog embryo if it lays eggs, and it's going to lay eggs. And so they actually were disseminated to hospitals as a pregnancy test. Well, there's a lot of biologists in hospitals. They said, wait, I can get eggs anytime I want just by going down there? And so it actually became an easy way to get amphibian embryos. And since then, it's been used for a huge number of discoveries. I don't know if you know, this year's Lasker Award was given to John Gurdon, among others, for his discovery using Xenopus embryos that somatic nuclei are actually totipotential. That is, they have all of the genetic information to make the entire organism. And that was found in Xenopus. The first eukaryotic genes were Isolated in Xenopus, mitochondrial DNA was discovered, nuclear targeting for proteins, discovery of the first Hox gene in vertebrates, all kinds of things. So Xenopus has been used for quite a while for a lot of different stuff, and it's what we use mostly in our lab. But what we're interested in fundamentally is the genetic process of the formation of the central nervous system. Now, if you're a neurobiologist, the question of how your brain gets made, and it's an immensely complicated question of how the different neurons all link up, and suddenly you can come up with E equals MC squared in these kinds of things, or you can decide to study amphibian gastrulation or whatever. But as a morphogeneticist, someone who's interested in how shapes are generated in embryos, this is how the brain gets made. You all know that your brain and spinal cord are hollow. They're filled with cerebrospinal fluid. Anyone who's had or has had a child who's had a spinal tap, what they're actually doing is sticking a little needle into that hollow tube and take some stuff out to see if there's infection in there. And so what you may not know is that your brain and your spinal cord were initially formed as a flat sheet of cells. And this flat sheet of cells rolled up into a tube, and in fact, one of your colleagues, Tim Green here, said, oh, it's like the tortillas and the fajitas you all had for lunch. That's exactly right. You got a flat sheet of cells. It's going to roll up into a tube. So this is a salamander embryo. That's another salamander embryo. There's two of them here. This part here at the top, I'll try to use the laser pointer, I guess. This part here at the top is going to be the brain, right here. This part down at the bottom is going to be the spinal cord. You're looking at about 6 hours in the life of a frog or a salamander. You did this. It took you a week to do this. You started doing this about 3 weeks after you were conceived. So 3 weeks between the talk before lunch and this talk. And you, come on, that was a pretty funny joke, right? Sex joke and conceived and, all right, okay, I'm sorry. You guys all fell for what I use on my undergrads. Is if you cop to the bad joke, you'll get a laugh, and then you get reinforcement. Okay. So this movie, it's fun to watch, but it also illustrates why we use amphibian embryos. This is very easy. We take these embryos, we stick them on top of a microscope, we can make movies with them. If we stick them on top of a more expensive microscope, we can make really nice movies of them. And now we can actually see that embryos are made out of cells, and that if a tissue is moving, it's doing so because all the cells within that tissue are moving in a really coordinated way to bring about this massive reorganization of this sheet of cells. It's going to close up, form a tube, and that's the central nervous system. Now, of course, you all know sometimes things get wrong. This is an embryo where we've done some genetic manipulations. I won't bother to tell you the details, but what you can see is the good side over there on the far side is rolling up quite nicely. Over here, however, nothing's happening. Okay? And so what we've done here is to mutate a gene that's essential for neural tube closure, and we've been studying those genes for some time in my laboratory. Now, why is this relevant? This is relevant because a defect in neural tube closure is among the most common, in fact, the second most common human birth defect are neural tube defects. These are things like spina bifida and anencephaly. And what these defects are, for the most part, are failures of this process, failure of the sheet to roll up and form a tube and seal shut. And so, you know, in my heart of hearts, I am fundamentally interested in how frogs close their neural tube. But I think it's also relevant that what we are studying is directly relevant to sort of the basic biology of a common human birth defect. All right, and so that's really the major focus of the lab. And what I'm going to take you through today is a really fun project that we've been going through the past 4 years or so that's led us to learn a whole lot about asthma, starting from this neural tube closure defect question that we were asking. Now, before I can get into that, I'm going to give you one really bad slide, and I'm sorry. These are all gene names. Every word below here is the name of a gene. And well, it's the name of some processes. And what I need to tell you is that signaling pathways control how cells behave. That is to say, proteins talk to each other, and by talking to each other, they can control what cells do. Okay? So there's two different pathways you need to know about. You don't need to know all the gene names. One is called hedgehog. One is called planar cell polarity. So only two things you need to know here. What hedgehog does is it tells cells, who am I? Am I a brain cell? Am I a spinal cord cell? Am I a kidney cell? This is what we call cell fate specification. It controls the turning on and turning off of genes in the nucleus. It's answering the question, who am I? Okay? This one over here, it doesn't say who am I, it says where am I. It tells cells where's the front, where's the back, where's the top, where's the bottom. Where am I, right? That's what these two different pathways are doing. And these are pathways that we've been studying. Really, this pathway over here, this where am I issue, has been the major issue that my lab's been focusing on. And we kind of stumbled into this, and we'll tell you a little bit about that. So we were going along, and we were making manipulations in these different genes, like that movie I showed you, and we caused neural tube closure to be screwed up. And you can see here's a normal embryo here and another normal embryo here. And you can see the embryos right next to them are not normal, right? There's a big hole in the back right here. That's an open neural tube problem. Here there's a big hole in the back also. That's an open neural tube problem. But if you're savvy, or if you just stare at it for a long time, you don't have to be savvy at all, you'll see that this is open up here at the top. This one's open down here in the middle. And really what this is this sort of red area. This is the spinal cord. This is a problem with the spinal cord. This up here is the brain. That's a problem with the brain. Now we know that human patients with birth defects tend to present with either brain defects or spinal cord defects, not generally both. And so the same in the mouse models, same in our frog models. And this didn't make any sense because every gene we had looked at in the PCP pathway, and we've looked at a lot of them, looked like this. But this guy looked like that. It looked different, okay? And that's really where we started from. This didn't make a whole lot of sense. And we wanted to go and ask some more questions about it. So when we did, we went looking at a whole bunch of phenotypes. This is a whole set of pictures you can ignore entirely. All you need to know is the top row. This is a phenotype. That's a characteristic outcome of manipulating the PCP pathway. So it did look like it was involved in PCP, like it's supposed to be. These guys down here, don't worry about what they are. These are eyes that are set close together. These are the jaws, and they're narrower compared to a normal embryo. These are all normal on this side. These are all ones where we've manipulated these genes. These are hedgehog phenotypes. Okay? So we did this in the mouse. We published this in Nature Genetics. People weren't entirely convinced by that, so then we did it in the mouse. And we knocked the gene out of the mouse, saw the same thing. It's hard to do the comparison, but here's that open anterior neural tube. Here's the jaw defects. These things are basically all saying that, yeah, there are defects in both of these pathways. But there really shouldn't be. These are genes that should only be involved in this PCP pathway. These genes should only be necessary for cells to know where they are. They shouldn't be necessary for cells to know who they are. And this little panel down here is this gene expression data. It says these cells don't know who they are. And this was quite unexpected. And so I'm going to sort of scoop myself here and tell you it turns out to be all about cilia. Cilia are hot. They were in the New York Times in May. I'll give you the reference to that article in a little while. So let me tell you what they are. I'm actually quoted in Nature about a year ago saying if you told me 5 years from now I'd be working on, that I'd be one of the experts, I guess, not the experts, an expert on cilia, I'd have laughed in your face. And it's absolutely true. These things are little projections that cells send out. They're filled with microtubules. That's a cytoskeletal element. Some of them move, and in fact, they were originally thought to be mostly involved in cells swimming. But some are actually non-modal. And in fact, the cell biologists will tell you, of course, most cells have one cilium, and it's non-modal. These things have been loved by cell biologists, but have been spurned by developmental biologists as vestigial. Okay? Until recently, okay? And this is sort of where the story goes from here, and we've learned a lot about cilia recently, and it's turned out to sort of hook all this together. So here's sort of the odd journey that I'll take you on today. We're going to start with these unexpected phenotypes, which really started from this gene expression pattern, and I'll explain this in a second. And we did some experiments, and we were developmental biologists. This is my training. I'm a developmental biologist. We went and we asked questions about how the embryo develops. Didn't make any sense to us at all. There's a lot of reading. You could also put right here where it's blank, you could say lost in the woods for 2+ years. Then we got a grant from an asthma foundation because we thought maybe this had something to do with cilia. We did more experiments, became cell biologists. We've been doing cell biology in intact embryos, which I think is pretty cool. We've published some papers, including this month's Nature Genetics, got the cover. I'm only putting this here because my undergraduate took this picture, and I'm always excited when undergraduates take pictures that get on the cover of fancy journals, so that's pretty exciting for us. We're doing more experiments, and really sort of who knows where it's all going to go. So, what is this? This is a frog embryo. You're looking down. It's sort of the same I showed you in those movies. This sort of white space here is where that neural tube is closing up. And what we've done to this embryo is to manipulate it in such a way that we can tell where a certain gene is turned on. We all have 20,000 genes. All your cells in your body have the same 20,000 genes. It's just which ones are turned on, meaning which ones are making RNA at a given time and which ones are not. This just tells us where the genes are turned on. Here's this gene fuzz that's been, or fuzzy, which is one of the genes we've been looking at. And here it is right in the middle of this neural tube. This made perfect sense. This is right where it needs to be for this neural tube closure phenotype that we're so excited about. But you can't help but notice all these spots, right? They're kind of pretty. And if you're a frogologist like myself, I've been working with Xenopus for 20 years. I knew exactly what they were, but I hadn't really thought much about them. They're ciliated cells. So if you go in with a fancy microscope, you look at the epidermis over here, you'll see that the epidermis of amphibian embryos at this stage covered with this red are just marking cilia. So these are these interspersed cells that make a whole bunch of cilia. And I kind of knew those things were there. I didn't think too much about it. Okay, yeah, it's in the ciliated cells, whatever. This is the meat right here. This is what we're going to look at. But then a funny thing happened. I started reading a little bit more because I think if you've got a phenotype, if you've got a situation, you don't know what's going on as a scientist, you should read. And when I start reading, I go all the way back. That's where I like to go first. So this is Richard Ashton's wonderful paper in the Quarterly Journal of Microscopical Science, 1896. And in 1896, it had already been frequently noticed. The fact that the embryo and larva of amphibia possess ciliated ectoderms has been frequently noticed. But there's been no description. My notes upon the ciliation may be of interest. So I started reading all this old literature and really thinking about these ciliated cells. And this has led to us learning a whole bunch of really neat stuff. And so at the same time I was kind of beginning to read up on these ciliated cells and think about what all this stuff might be doing, was the same period of time when this explosion in cilia research came about. And for you guys, what you might want to read is this one by Wallace Raven, New York Times, May 18th of 2009, Antenna on the Cell Surface, the Key to Development and Disease. This is a wonderful popular piece on the recent explosion of cilia research, and I recommend it to you. But the science behind it, I think there's been several papers. Really, the beginning was this paper from Catherine Anderson and Lee Neiswander's lab, where they showed, and all this stuff, basically that cilia were necessary for that hedgehog signaling pathway. Remember, that's the pathway I told you about that controls who a cell is going to be, okay? And that there was some link between cilia and hedgehog. And this is another follow-up, and there are lots of follow-ups. I put this one because Jeremy Ryder is a friend of mine, and it was while having a beer with Jeremy Ryder at a meeting and explaining my very peculiar results that these PCP genes seem to be having these hedgehog defects that we actually said, we started talking about the cilium and made the connection to that gene expression in the ciliated cells that made me go off and look. And in fact, when we go do some analysis here, are these multiciliated cells are making hundreds of cilia, and here's when we knock this gene down, the cilia are just gone. Same thing in the mouse. These little red things are cilia, and you can see that they're quite a bit shorter here in the mouse. And so what we found that was kind of really surprising, there was no evidence that these two pathways should talk to each other. And it turns out they talk to each other in a very indirect way. That is, you've got this signaling pathway that's doing something down here, but that's necessary for ciliogenesis. Because ciliogenesis is necessary for this hedgehog pathway, you have these neural tube closure defects. So it's a very indirect path that's taken there to get these phenotypes. Now, this is interesting because it tells us a little something about sort of the molecular biology of this particular neural tube closure defect. And we were pretty excited about that, but kind of couldn't get over this other tissue, this ciliated epidermis. I was really enchanted with it. These are some data from Richard Ashton's paper, again, 1896. Notes upon the ciliation of the tadpoles may perhaps be of interest. He did a couple of cool things. One, he cut a cross-section, and he looked down the microscope, and he drew what he saw. There were no digital cameras in 1896. And he drew what he saw. And, here are these multiciliated cells sitting here, interspersed with these other cells, sort of unknown function. But what really caught my eye was this picture here. What he did was to drop charcoal particles on top of tadpoles. He would grind up charcoal into tiny little particles and drop them. And then he noticed that there was fluid flow across the surface. He wasn't the first to do this, but as near as I can tell, he's the first to really map out the fluid flow. And that was that these cilia were all beating in the same direction. Okay? They were moving fluid across the surface of the tadpole in a very orderly fashion from the front of the tadpole to the back of the tadpole. So this was interesting, and this has actually been studied in beautiful papers from Victor Tweed in the 40s, another paper in the 80s from a guy who's named Peter Hausen, who's only recently retired. And some people were working on this, but not much, and I was thinking quite a bit about this at the time. And it occurred to me that there seemed to be a similarity between this tissue, where you have these multiciliated cells in these other tissue, and it called to mind what's going on in the airway, actually. So in your airway, you have exactly this same setup. You have multiciliated cells that are interspersed in the lining of the airway with mucus-secreting cells called goblet cells. Don't worry about these cells over here. So you have these mucus-secreting goblet cells, and you have these ciliated cells. And what happens is the mucus is actually the first line of defense. Every time you take a breath, billions of bacteria, maybe some viruses, lots of particles, lots of all kinds of bad stuff that would hurt those cells that are necessary to do the gas exchange, right? So you can breathe, right? But every time you breathe, you get that stuff. So the way you protect yourself from this is with mucus. Everybody hates mucus because when you get sick, it makes it really hard to breathe and annoying, but you couldn't live without it. You'd be dead in a few weeks. You know, you'd have infections. I don't know if you'd be dead in a few weeks. You wouldn't last long. I'm prone to hyperbole. I apologize. But the way this system works is actually really clever. If you just made a whole bunch of mucus in your lungs, you'd drown in it, as we all know from when we get sick. And so what happens is you have these multiciliated cells. They're all beating in the same direction. By beating in the same direction, when you're making this new mucus, the stuff all goes up to your oropharynx, and you're constantly swallowing. All day and all night, you're constantly swallowing small amounts of mucus that are getting there, not because you cough. When you're sick, you cough. I'm actually getting over something. I have 2 small children, so I'm very familiar with my mucociliary epithelia right now. But what happens is you're not coughing all the time. These cilia are beating, they're moving that mucus up. This is exactly the same thing as what's going on here, it seemed to me. And so then I started thinking about it, and I thought that was kind of exciting. And then I wasn't really excited until I got an advertisement. I was a victim of a cold call, actually a mass circular. I was given something in the mail that said, have you never ever thought you'd work on asthma, but you have a good idea? We'd like to give you a lot of money to pursue that. This is from a wonderful group they're now called the American Asthma Foundation. They used to be called the Sandler Program for Asthma Research. They give grants that are very large. Even the ones to the young investigators like myself are very large. And you can only get this grant once, and you can only get it if you've never worked on asthma before. And they like to only give it to you if you're doing something kind of ridiculous, as near as I can tell, because there's no other reason to explain why I got an asthma grant. But I saw that, and I said, well, maybe I'll learn a little bit. And so then I went to the library. My undergrads, even my graduate students don't really understand why you would go to the library to read, but I find it peaceful. And I went to the library and started reading. And I actually found out that there was a lot of biology left to be learned here. And when you think about it, it makes a lot of sense. Why is it that we know, from a biology standpoint, we know very little how these tissues develop? How does this guy know he's going to be a ciliated cell? How does this guy know he's going to be a mucus-secreting cell? That's a who I am question, right? How do they know to build the cilia up here and not up here? That's a where I am question. There were all these kinds of questions that I'm really interested in. I thought that's very exciting. And I went reading, it turns out we don't have any idea about this, and then it occurs to me that probably the reason we don't know much about it is this is a process that happened inside the tiny little airways of the embryo, and in a mouse, where this kind of thing is usually studied, it happens inside mom. So you're inside an embryo that's inside a uterus and inside something that's crawling around. So this is a very intractable tissue. It's very hard to study, and so it hasn't been studied very well. But what we do know is that defects in this mucociliary system are rampant with asthma and also most other airway pathologies, COPD, cystic fibrosis, a number of these things. You see a lot of broken cilia that don't beat with the right frequency, they don't beat in the right direction. It's not really clear why. You also get this overproduction of mucus. This is a real problem, and in fact, in an asthma attack, generally what's going on is your airway is getting smaller because the smooth muscles around the airway are constricting. But what makes the difference between an asthma attack and a fatal asthma attack can sometimes be mucus plugging. It's a horrible term. It's called fatal mucus plugging. If you have a lot of mucus hypersecretion, when your airway gets small, you're not able to tolerate that excess mucus. It plugs your airway completely. This can turn an asthma attack into a fatal asthma attack. Not a whole lot of biology is known about this. And so we thought, well, maybe we can model airway pathology using an animal with gills. See, gills are not lungs. Come on, this is a great joke. We're going to try it again, all right? They got gills. They don't have lungs. They're tadpoles. They're amphibians. They're swimming around in the water, and yet they've built this beautiful mucociliary epithelium on their epidermis, right? It's a courtesy to biologists, I think, that they've done this. You could make evolutionary arguments, but it's funnier to say it that way. But what's really nice about this is, as I've told you before, these are externally developing animals, right? They just poop in a dish of water. Anyone with kids has gone and found tadpoles in a pond. You know these things are there. So it's not just that. It's not just that the embryo develops externally, but that this mucociliary epithelium is actually on the surface of the embryo. It couldn't be easier to study. So is it really like the airway? Well, it turns out that yes, it is. Studies from several labs, including my own, we know that it has a very similar morphology. It has a similar ultrastructure. That means if we go in and we look at by electron microscopy, it looks very much the same. They secrete mucus. They have oriented surface flow. And what we've been doing recently, as well as a couple of other labs, one at the Salk Institute, one now at Northwestern University, finding that the same genes are expressed in the ciliated cells of the frog epidermis as are expressed in the ciliated cells of the human airway. The same genes are expressed in the mucus of the frog epidermis as are expressed in the mucus-secreting cells of the epidermis. So this interleukin-2 gene is actually highly overexpressed in asthmatic patients, and it's present in these guys. We've actually found that the molecular signaling pathways that control the spacing in number of these ciliated cells, 2 beautiful papers came out on the mouse showing that this pathway called Notch controls this process. Well, we've actually known that in Xenopus for about 10 years now. So it turns out to be a great system. We can go in, we can do all kinds of imaging to look at these beautiful things. And so we've been doing that. So what I'd like to do in the next couple of minutes now is to just show you some ways that we can model these processes that are normally completely invisible because they're inside the airways of a mouse that's inside the uterus, and we're able to just go and look at it. So can we watch cilia-mediated fluid flow? Well, I told you that 120 years ago, people were already dropping charcoal particles on and drawing pictures. Well, we can go one better. This is the side of a frog embryo. Here's what the frog embryo would look like. We've stained it here to look at the ciliated cells. You won't see the ciliated cells here, but that's the flank of the embryo. We've got a needle here. The needle's going to drop out these blue latex beads, and when the movie starts, you're going to see them flow along. And so not only can we see the fluid flow visualized this way, we can actually measure it. So this is for effect. We're putting in tons of beads and watching them go. If we use a small number of beads, we can actually trace the direction and the velocity of those beads and actually learn a lot, really sort of quantitative data about fluid flow. So we can go in, we can manipulate the embryo and quantitatively assess fluid flow in this surface, okay? What about mucus secretion? Yes, we can. So this is a fluorescent version, basically a glowing version of a human mucus protein, which we've put ectopically into this frog embryo. And you can see that it's actually packaged into these nice little granules, which are these mucus-secreting vesicles here. And when the arrow comes up, we're actually going to show you a couple of these are going to go away, and this guy's going to we can actually watch in real time the secretion of mucus in this tissue. And this experiment was done by a rotation student who had just started grad school, had been in the lab 2 weeks and did this experiment. Couldn't be easier. Now, the airway doesn't only contain these mucus-secreting cells and these ciliated cells. In fact, there's quite a few immune cells that are floating around in there. This is a beautiful work. It's not my work at all, from this Veraz et al. Paper. And what they've done is they've actually gone in and they've sort of virtually dissected the airway. So the purple here is showing you where the airway is. And what they're showing you in yellow is these dendritic cells. These are immune cells that are patrolling the airway. And so does our tissue have that as well? It turns out they do. In fact, we can watch them being born. This is a population of immune cells that are going to be born about this time as the embryo is developing. And they're going to do something wonderful. They're going to just migrate out of here. They're going to populate the entire epidermis. And then they're going to patrol around looking for bugs that they can eat. They're going to respond to wounds. They're going to do all kinds of things. And again, a simple experiment to do. And so really, when we look at this tadpole epidermis, we realize that it almost is the lining of the lungs on the outside of a tadpole. It makes it very, very easy for us to go in and do experiments and learn things about how these systems work. So in the next set of slides, real quick, I'm just going to sort of tell you a little bit about the things that we've learned, and then show you a few more movies, and we'll get out of here. So one of the new things that's going on that's wonderful is the ability of bioinformatics to predict protein function. Now this is really nice because this gene we were studying, fuzzy, and this isn't the only one. This is just the example I'm using for today's talk. This is a gene that we're really excited about, but it's entirely novel. It doesn't look like other genes. A lot of times you find a gene, it looks like some other gene, it gives you some idea, oh, it's an enzyme, okay, it's going to be kind of like this other enzyme. You can actually make some hypotheses about what it should do, and it can guide your research. Well, fuzzy doesn't have that advantage. It doesn't look like anything else. However, through bioinformatics, you can make predictions about it. Now, one of the problems with coupling bioinformatics to the mouse is then if you want to go manipulate it in the mouse, sometimes it can take months, sometimes it can take years, it's very expensive. The frog is cheap, it's fast, it's easy, we can go through very quickly. So what we've been doing recently is focusing on using bioinformatics to predict the function for proteins and then testing these functions very quickly using this ciliated epidermis. And one of the things we found was that fuzzies seem to be involved in vesicle trafficking, that's secretion basically, and really all you can see is that here are these pits that are opening up, they're going to release this mucus. And what you can see is these pits never open up when we disrupt this gene. And so not only do you have the cilia defect, but you also have this failure to secrete mucus. And so this is a novel insight for some kind of link between secretion and ciliogenesis. We don't know what that is, but we're working on it now. We can also predict co-conspirators this way. So what you're seeing here in green is another protein called clamp. Don't worry about it, what it does. All you need to know about is where it goes. So here's one of those multiciliated cells, and what you can see is clamp actually gets stuck out at the ends of all these cilia, right? I think this is a really pretty picture, and you can go, we've made a mosaic here where this is a normal cell, but this is a cell where we've disrupted the function of fuzzy. You can see they're making these sort of mimic-looking cilia, but what's most notable is none of this bright green spots at the ends, the tips of these cilia are present in this cell. And so it seems that fuzzy's somehow working together with this protein clamp to cause this ciliogenesis defect. We've also been able to use this to do gene discovery. So there are actually massive databases of where genes are expressed. I told you that every cell has all the same genes. It's just which ones are turned on and which ones are turned off. And so we can use this tech that I showed you before. Here's these spots like I showed you for fuzzy. That's the ciliated cells. Well, there are databases of these pictures. So I spent a weekend. I don't do a whole lot of experiments. All the data that I'm showing you, some of the ideas are mine, but all the data is coming from the students in my lab. But I actually did this, and I'm actually very pleased because I did it while I was watching a football game. I sat at a database while all day Sunday, kids were out of town with their mom at their grandparents. That's why I could watch football all day long. And watch football all day long. I don't even like football that much, but when the kids are gone, right? So watch football all day long, surfed databases of gene expression looking for things that had white spots on a black background. That should be the mucus secreting cells. Dark spots on a white background. And in a weekend, identified over 150 differentially expressed genes, many of which are relevant for asthma and other airway pathologies. So, the last sort of thing that we've learned, again, there's some problems with building these cilia. What about that directional beating? I'm going to skip some of the details. This is a different gene. We've manipulated it a different way. But these fluid flow diagrams from 100 years ago can actually be pretty interesting. So here's a movie where we've put in just a few beads, and I hope you can see these little spots are moving basically from left to right on the screen, and they're flowing kind of like this, left to right and up to down, kind of like what you saw in these arrows right here. And when we go in and we manipulate this gene, you can see here, hopefully you'll be able to see the movie, but they're kind of spinning in a little vortex right here. And by doing this type of analysis, we're actually able to get quantitative data on exactly what's wrong with the flow field and how has the directional beating of these things been screwed up. And you can imagine that this also would be relevant to the clearance of mucus in the airway. And finally, we've been able to go in most recently and actually look at these cilia. And we've now got an even more expensive microscope that goes at about 370 frames per second. So these cilia beat about 20 times per second. We're able to watch this as a single multiciliated cell. These are dozens of cilia, and they're all beating. And what's interesting is they're all beating at the same time. They're all beating roughly in the same direction. We can watch this because, again, it's on the outer surface of this externally developing animal. When we go in and we manipulate, as we did before, the way that disrupts the fluid flow, you can see everything's gone haywire. They're not beating at the same time. They're not beating in the same direction. In fact, they look a little bit stiffer. They don't look like they're bending as they're going. There's new evidence that bending is related to the directional flow, these kinds of things. And so this is a whole new area of understanding how cilia beat and how they beat directionally. And so where we started was just this observation that cilia were somehow linking these two signaling pathways. And these are signaling pathways that are heavily studied by lots and lots of scientists. This is the first connection between the two, and it's through this really indirect way. And what we've been able to do very, very rapidly is to fill in lots of the details here. We've been able to learn a huge amount about the protein networks that are controlling ciliogenesis. They're also controlling secretion. The ciliogenesis is necessary for this hedgehog signaling, also for directional beating. We've basically been able to do a huge amount of biology on these mucociliary epithelia, again, simply by being a little bit curious about why the frog embryo develops the way it does and how we might leverage that to learn some new things. And so finally, just to wrap up, what did we learn? We learned that frogs are cool. We learned that you should read the old papers. We've made new links between important signaling pathways. We developed some new tools for studying mucociliary epithelia, and most importantly, we've done this in vivo. So a lot of this type of work requires cell culture. You take the cells out, you put them in a dish. Well, cells out on a dish, that's not how they're supposed to be. They probably behave a little bit differently than they do when they're in their native environment, so we're able to do these things in vivo. We've found some new genes, and I sort of have the same thought about genome-wide association studies, and so we go a different way. We sort of take a candidate gene approach. All the genes that we found are probably relevant to birth defects and to airway pathologies. How this translates into clinical relevance, we have no idea, but I'll tell you it is great for maintaining childlike exuberance, which in my business is incredibly important. And so then the last slide, I'm sure I'm going to get this question, so I'm going to answer it now. Why on earth do they have these things on the surface of the epidermis? Why have they recreated the lung on the outside? Well, so there are a lot of different thoughts about this. There's a wonderful paper from this guy, and I dare you to pronounce his name. I've never even tried in public to pronounce his name. What I love is that he's at the University of Shiraz in Iran, so we're doing some diplomacy here. I will not talk to him without preconditions. I will now discuss our research without preconditions. But basically, he comes up with several. He's very circumspect, and I think that's a good thing. But basically, what the thinking is that these during these early stages, a lot of the respiration that the tadpole does is gas exchange through the epidermis, and that by moving fluid across it more rapidly, especially when they're clustered together, as amphibian embryos often are, is it increases oxygen flow to the region by moving the fluid, and actually is a respiratory function, although there are a lot of other possibilities as well. And so finally, I just really want to point this out. If you hear some guy stand up at the front and drone on and on and on about how great the research is, it's usually not something they've actually done. It's usually something someone else has done. And so all of this work was done by people in my laboratory. Teju Park and Ryan Gray are graduate students. Phil Abatua was the undergraduate whose picture ended up on the cover. And I guess Julie Hayes is not in this picture, and Julie Hayes was also heavily involved in Emily Harrington in the differential gene expression studies, and then all these collaborators and these people who gave me money, and thanks to all of you guys for listening.
You got the first question last.
Okay, well, I'll be very quick. It's fascinating work, but I'm confused about some terminology.
I am too.
Well, I thought Xenopus were toads and rans were frogs. And I noticed in the old, has the terminology changed because Heatherton or whatever his name is?
Right. So I get this question frequently, and I have to tell you that my early attempts at field biology were met with disaster. It's the consensus in the field that of people who do biomedical research with Xenopus, we call them frogs. A naturalist, a proper evolutionary biologist, might be able to say something different about the classification as to whether they're frogs or toads, but we call them frogs, and I just don't honestly know the truth.
Okay,
That's a terrible question to get first. I don't know. Sorry.
So this is kind of off topic, but how exactly was it discovered that urine from pregnant women induced the Xenopus to lay eggs?
Wow. This is a tough audience. I don't know. I would like to say childlike exuberance.
Do we have any other questions, any sort of more direct questions? No? Well, I guess you answered them all in the talk. This is
Outstanding.
Fantastic. Okay.
I've
thank
done my
you.
job well. I've got you guys back
Wait,
on time,
wait.
too.
Oops, we got one. We got one.
Sure.
I'm wondering if this research could also translate to the intestinal tract or not, or if that's too, if
Right,
villi
so
and microvilli are
that's
close
exactly
enough
right.
to that.
So these types of ciliated cells are not going to be present in the intestinal tract. We think some of the secretion stuff is probably likely. Where it is probably going to translate is the fallopian tube. Okay. So it's actually multiciliated cells are what move eggs along the fallopian tube. And in fact, people who have what's called primary ciliary dyskinesia are often infertile for this reason. And in fact, there is some of the people who do research on directional beating, it's actually recently been shown, I saw at a meeting, it's not published, these PCP genes are also necessary for directional beating in the fallopian tube of mice. And so absolutely it'll translate to the fallopian tube.
Okay, thank you again
Thank
very
you.
much.
Pleasure.
And we'll